Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning. Today we will focus on fungal symbiosis, specifically mycorrhizal relationships with plants, and why these partnerships matter in ecosystems and agriculture. Mycorrhizae are intimate associations between fungal hyphae and plant roots. There are two broad types that we encounter most often. Arbuscular mycorrhizae form tiny branched structures, called arbuscules, inside root cortical cells. Ectomycorrhizae form an external fungal mantle and a heartig net between root cells rather than inside them. Arbuscular mycorrhizae are common in herbaceous plants and many crops. The fungus belongs to a group now usually called Glomeromycota. One well-studied species is Rhizophagus irregularis. In field trials, inoculated wheat and maize often show increased phosphorus uptake when colonized by this fungus. Ectomycorrhizae are typical of many trees, including pines and oaks. Species in genera, such as Sualis and Pisolithus, form visible mantles and provide their hosts with nitrogen and water while receiving carbon in return. At the cellular level, arbuscules increase the surface area for nutrient exchange. The plant supplies sugars and lipids to the fungus, and the fungus supplies minerals, especially phosphorus, and sometimes nitrogen derived from organic matter. Plants may pay up to 20% of their photosynthate to support mycorrhizal fungi, a substantial carbon cost. That cost is balanced by improved nutrient acquisition, better drought tolerance, and enhanced pathogen resistance in many cases. I often say fungi are not only nutrient scavengers, but also gardeners of the rhizosphere. By altering soil structure, producing enzymes, and shaping bacterial communities, they change the immediate environment around roots. A fascinating phenomenon is the common mycorrhizal network. Individual fungal mycelia can connect multiple plants and shuttle carbon, nitrogen, and signaling molecules between them. Isotope labeling experiments using 13C and 15N show movement of these elements along hyphal pathways. These networks can facilitate cooperation when, for example, shaded seedlings receive carbon from well-lit adults, but they can also mediate competition if dominant plants draw resources away from neighbors. Specificity varies. Some mycorrhizal fungi colonize many plant species, while others are host-specific. Orchid mycorrhizae are highly specific and critical for orchid seed germination. Without the right fungal partner, an orchid seed will fail to develop. Molecular signaling initiates colonization. Plant roots secrete strigolactones that stimulate fungal hyphal branching. In response, fungi produce MYC factors that trigger cortical cell responses and arbuscule formation. From a practical standpoint, mycorrhizal inoculation is used in restoration and sustainable agriculture. In degraded soils, inoculation with Pisolithus or Rhizophagus can increase survival of seedlings and reduce need for phosphate fertilizers. However, inoculation is not a universal solution. Soil conditions, native microbial communities, and plant genetics influence outcomes. Introducing non-native fungal strains can have unintended ecological consequences. Research methods combine microscopy to identify arbuscules and hardig nets with molecular tools to identify fungal taxa. Stable isotope tracing demonstrates resource flows. Field trials then evaluate growth and nutrient responses. In summary, fungal symbiosis encompasses diverse interactions from strict mutualism to conditional outcomes. Understanding signaling, carbon costs, nutrient exchange, and network dynamics is essential for both ecology and applied management of plant communities. That concludes the lecture. For our next class, we will examine specific case studies of mycorrhizal restoration in mine spoils and urban soils.
Listen again to part of a lecture, then answer the question.
Common mycorrhizal networks can shuttle carbon, nitrogen, and signaling molecules between plants.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.